My name is Emily Fisher, and I'm going to be talking to you today about how Wnt beta-catenin signaling drives age-dependent oral squamous cell carcinoma evolution by taking you through my electronic poster. I'll discuss the background of this disease, the materials and methods I use to study this disease, as well as the results and conclusions of my experiments. Head and neck squamous cell carcinoma is the 7th most common cancer worldwide, with over 900,000 new cases diagnosed annually. Oral squamous cell carcinoma is the most common anatomical subsite of HNSCC cases. The focus of my research is on HPV-negative oral squamous cell carcinoma, whose etiology includes tobacco use and alcohol consumption. Age is an acknowledged risk factor for HPV-negative OSCC, with the median age of diagnosis being 66 years. However, the majority of research is done in young patients or animals or using in vitro studies such as cell lines, none of which capture the age effects that may be contributing to OSCC tumorigenesis. Our lab and others have shown that OSCC tumorigenesis is driven by canonical Wnt signaling. Specifically in this disease, beta-catenin translocates from the cytoplasm to the nucleus, where it interacts with the transcriptional coactivator CBP, or CREB binding protein, resulting in the transcription of genes associated with stemness and partial epithelial to mesenchymal transition. Furthermore, CBP is able to recruit MLL1 to the transcription start site of these genes. MLL1, or mixed lineage leukemia 1, is a methyltransferase that is able to establish open chromatin through trimethylation of histone 3 lysine 4, collectively reinforcing this transcriptional program of beta-catenin and CBP. To perform functional studies of this pathway, our lab uses E7386, a third-generation small molecule inhibitor that binds directly to the N-terminus of CBP in a reversible competitive manner, preventing beta-catenin from binding to CBP. I use 3 different age cohorts of mice to study OSCC in vivo: young mice, middle-aged mice, and old mice. Referencing this diagram on the right, which comes from Jackson Labs and is a mouse to human age conversion, we can see that the target population of patients who are diagnosed with OSCC at 66 years correlates with my old mice or grandma mice age cohort. Young mice are the ones that are most commonly used, however, they're only about 20 to 30 years in human years. In the foreign QO-induced model of oral carcinogenesis, mice are exposed to foreign QO, a synthetic carcinogen that mimics the carcinogens found in tobacco, and mice drink foreign QO in their water for up to 4 months. OSC develops by week 25 in this model, so approximately 6 months in. In 2019, Dr. Goodkin published a 4MOS1 isograph model, which involves injecting cancer cells isolated from mouse foreign QO lesions directly into the tongues of C57BL6 mice. The resulting tumors have approximately a 94% mutational signature overlap with the human disease, making it a powerful, accurate, and much more time-friendly experimental model than the 4O and QO-induced model. As you can see here, tumors are able to develop in a matter of days rather than months with the 4MOS1 isograph model. However, I do use both for my studies. Characterization of the 4MOS1 cells using a top FOP assay show that they are responsive to Wnt beta-catenin signaling, as evidenced by the increased luciferase activity in the top transfected and top transfected plus 40 millimolars of lithium chloride treated conditions, compared to the scrambled control FOP condition. The majority of Wnt beta-catenin signaling was abolished upon treatment with 1 micromolar of E7386 for all conditions. Furthermore, a Presto Blue cell viability assay also shows my 4MOS1 cells display a dose-dependent decrease in cell viability upon treatment with E7386, confirming they are responsive to Wnt beta-catenin CBP signaling. Further in vitro characterization of E7386 shows a decrease in CBP protein across two different human cell lines of OSCC, Cal27 and HSC3. MLL1 and its epigenetic mark, histone 3 lysine 4 trimethylation, are also decreased upon treatment with E7386 as compared to their DMSO controls. Significantly, beta-catenin protein levels do not change upon treatment with E7386, which is important given the various functional roles it plays in cells. A ChIP-seq analysis of HSC3 cells show that upon treatment with E7386, we are specifically seeing reduced histone 3 lysine 4 trimethylation histone occupancy at the transcription start sites of cancer stem cell genes, including CD44 and ALDH7A1. In vivo analysis of young and middle-aged mice 4MOS1 isograft tumors show that old mice have increased CBP and histone 3 lysine 4 trimethylation expression and fluorescence intensity as compared to the young cohort of mice. This suggests that the beta-catenin CBP MLL1 axis may be upregulated with age. When we orthotopically inject the same number of 4MOS1 cells into the young and old mice tongues, we see that the old mice grow tumors much larger and much more quickly than the young mice. However, in the clinic, patient treatment is decided largely based on the tumor stage. Therefore, in my models for evaluating OSCC tumorigenesis, I stage my tumors by injecting fourfold fewer 4MOS1 cells into my old mice as compared to my young. This allows me to evaluate tumors that are roughly the same volume at the start of treatment in my young and old mice, allowing me to isolate simply how age affects tumor growth and reduction. As we can see, in my young mice, they have approximately a 70% reduction in tumor volume upon daily oral gavage treatment with E7386 for a total of 10 days. In our old mice, while we see a statistically significant reduction in tumor volume, it is not as robust as what's observed in the young mice, suggesting a more complicated microenvironment and pathogenesis of OSCC tumor growth. So I hypothesized that in addition to the beta-catenin CBP MLL1 signaling axis driving OSCC tumorigenesis, old mice also have age-associated factors, including the presence of senescent cells and a decreased immune function that promote tumor growth. Senescent cells increase with age. They are cells that have exited the cell cycle as a result of a variety of different stressors and adapt a pro-inflammatory phenotype known as the senescence-associated secretory phenotype, which has been shown to transform neighboring cells into pro-tumorigenic stem-like cells. Immunofluorescence analysis of my 4MOS1 isographs show that my aging mice tumors have increased markers of senescence, as noted with our loss of lamin B1 in the nuclear envelope, concomitant with increased histone 3 lysine 9 trimethylation epigenetic marks, which are required for the induction of senescence. I am in the process of further characterizing senescent cells in my young and old isograft models. Additionally, our single-cell sequencing results on the right show old mice tumors have fewer immune cells than our young mice, including neutrophils, antigen-presenting cells, and T cells. These cells are key in preventing tumor growth, and their absence in old mice may be responsible for the limited reduction in tumor volume we see upon treatment with E7386. In conclusion, epigenetic modifications directed by the beta-catenin CBP MLL1 complex promote expression of genes associated with cancer stemness. While OSCC is driven by the beta-catenin CBP MLL1 signaling axis, as evidenced by the tumor volume reduction upon treatment with E7386 in vivo, the microenvironment of older mice tumors may be more complex than what's seen in young mice. Increases in IF associated with senescent markers suggest the potential involvement of this signaling axis in this age-related pro-tumorigenic cellular phenotype. And finally, aged mice have fewer immune cells in the tumor microenvironment, potentially contributing to OSCC tumor growth.